Thanks, Bruce.
Good evening, everybody. Thank you for having us. It's a great honor to give this memorial lecture. Thank you, Bruce, for introducing us and for your stewardship and guardianship of the institution and for your vision of guiding our research. And thank you to all of you for joining us tonight and listening to our ideas and to the way by which we form research. We thought we'd do this in a bit of a conversational style, and we tell you a little bit how ideas like the ones that we are working on maybe emerge at Cold Spring Harbor, and then how we think about bringing them to the clinic. So, Lloyd, how do you remember that we got our collaboration going?
The funny thing I would say is that it kind of started during COVID. As Bruce was just mentioning, we were all hunkered down at home and had time to think about what turned out to be a huge problem, also scientifically. And then I hear and read that Cold Spring Harbor Lab is doing a trial. And I have, at that time I had been here for like 16 years or so I came in 2007, 19 years ago now. And I never heard of us doing a trial. And then obviously, also on the trial that's COVID related. So something that really was thought of instantaneously and executed. And so that's when I realized that you are actually an MD. He doesn't just have the MD PhD in his name. He actually knows how to get things into people.
Yeah, thank you. I remember, sort of from my perspective, we have at this institution very many opportunities to exchange ideas, and we share these ideas very freely, and that's really encouraged by the leadership and embraced by everybody at the institution. And Lloyd presented fascinating data in one of our in-house symposia way before it was published, so that other people could give critical feedback. And we were at that point trying to develop behavioral assays in our lab that would actually lend themselves to the tail end of your discovery, really to get a better understanding of what happened in the mice model. And we spoke afterwards and started collaborating actually through the research project first, and then starting to think more about patients. And today we want to give you a short summary of both of those events, starting with Lloyd's discovery work, and then I will tell you a little bit about how we reason how to bring this into clinic. And maybe the first slide, I always find that the appreciative if you contextualize it in history. You see there an image of Al-Razi, who was a Persian physician in the 9th century, and who maybe was the grandfather of translational science. So he was actually somebody who would have embraced the Cold Spring Harbor principle of doing experiments to find out what's best done. And I'll give you one example. For example, he was tasked with building a hospital in Baghdad, which was then an epicenter of scientific discovery. And the experiment that he did is he took fresh meat and put it out on sticks everywhere across the city and monitored where the meat would rot first and where it would rot last, and then decided to build the hospital in that spot where the meat was preserved for the longest. And so that was a real experimental medicine study informing hospital care of sorts, and in the tradition of experiments and leading into clinical trials. And Lloyd and I are trying to continue this on a slightly deeper level. And Lloyd is going to speak about the science first, and for props, he's going to wear a lab coat, I'm going to wear a hospital coat, and so that you know where we are at each point of the story.
And I thought we would make this real, and I show you the standard issue James laboratory coat. There are actually several colors depending on where exactly you're doing your experiments. And I'm not going to embarrass myself, and I can actually still put one of these on.
May I give you a hand? No.
Because it's been a while. So it did obviously all start in the lab. And what's important to this point is, of course, the people who have been on this journey. I'm going to summarize in about 5 slides some 10 years of work. We started this work exactly at the point that Bruce mentioned before, when we had developed the first mouse model that developed metastatic prostate cancer naturally, just like a human would, by a sort of bad luck incident. And that mouse model then prompted us to ask if we could do therapies that go way beyond what is standard of care today. Because that is a question you're not simply allowed to ask in humans. You have to give standard of care. So these are the people, and I would want to highlight Manojit, an extremely talented PhD student who is now doing his postdoc in a famous lab, who has taken on this study. And so most of the data and results that you're seeing started with him, but everything is only possible because of the entire team. So the question that we were asking at the time when we had the mouse model was the following. Prostate cancer is a disease that affects many men, but unlike many other cancer types, there is this huge gap between having the cancer and actually dying from it. So the chances of actually developing the lethal metastatic prostate cancer are just a few percent. So this leaves many men with either a diagnosis or perhaps the fear of being diagnosed later on to ask the question, what do I do to keep this at bay or keep it away for good? And the answer to that for many of us is the lifestyle. And with a lifestyle comes diet, but with diet also comes supplements. And as you can tell from this picture here, the supplement market, there's no shortage of supplements wherever you go. This is here on the East Coast. This is in China. There will be on any mall, there will be supplements. And obviously I can't read the price tags, but they are quite high. I can read this price tag. That is a picture taken from a store in Los Angeles that's now really, really trendy. And they're selling something that is also very trendy and also very important. That was the origin of the study. They're selling antioxidants. And I would assume that everybody in this room has heard of antioxidants and the benefits of antioxidants, et cetera, et cetera. And glutathione is a drug that we use in the lab, and I'll show you an experiment with it. And you can see the price tag is not to be underestimated. So antioxidants and their role in cancer. So the fact has been, and we knew that 10 years ago, that dietary antioxidants have been tested in prostate cancer. They've been tested on something like 23,000 men that were enrolled and randomly assigned daily antioxidants or a placebo. And then the question was asked, in which group would there be more prostate cancer patients prospectively? So which group would just by chance develop or would show more higher prostate cancer incidence. And of course, the expectation was that this could be now finally demonstrating the benefit that the antioxidants have in prostate cancer. But the opposite was the case. And it's the exact opposite. So you see two lines on this curve, and that's the probability or the numbers of men that develop prostate cancer. And that's, again, it's 35,000 men. The two groups were, there was a 20 and a 17,000 men group. And the follow-up was 10 years. So a very, very significant follow-up. The men who took vitamin E, a classic antioxidant, had a 17% increased risk over those who did not. So the exact opposite of what was expected and what we generally had thought of as antioxidant power. And so the big question for us scientists at the time when these results came out, and with the new model in hand, was, well, if the antioxidants could promote prostate cancer, would a pro-oxidant decrease prostate cancer? And that's, of course, the question that there wasn't any appetite for doing in men. The 35,000 enrollees and their follow-up cost a fortune. But that's where a model like we developed can really shine. So the model that we developed is shown here. We call it rapid cap. The disease starts in the prostate of the mouse, and we can image the animal without opening up or any, just like you would a patient, and the disease spreads. And then we can do standard of care, which here is castration, but it's anti-hormonal therapy. And you can see that that has a profound and can have a profound effect. However, that's only temporary. Standard of care will fail, and there's always relapse. This is the graphical annotation of these results. So there's always a relapse. And when we gave a pro-oxidant therapy, we saw that instead of the water where the disease would just progress and increase, we had many animals where the disease was simply subdued and did not come back. And so when we think about the pro-oxidant that we gave, we were actually quite surprised, because it is ultimately a dietary supplement / vitamin intermediate. So the vitamin biology, everybody knows vitamin K, which is present in plant greens mostly, or also in cheese. And that is transformed into the vitamin K that we have in our bodies. The intermediate in that transformation process is called menadione. It is produced from the vitamin K in the gut and then goes into circulation. So it's something that we are very familiar with. And this is the agent that we used that had the anti-cancer effect. Manojit and Shan, who's here in the audience, then were able to show how this works. When we give menadione, the agent, the pro-oxidant, to cells, you will see what happens here. So cells who aren't giving anything are simply dividing and living happily, you can see here. Cells who receive menadione, they die off. And they die off, as you can see here, in this very weird way. You see these large vacuoles that are appearing. So we figured out that this is an entirely new type of cell death that is induced only by menadione. And when you give the antioxidant is like an antidote. The cancer cells survive thanks to the antioxidant. So that was completely consistent with the notion that antioxidants can promote the cancer and the pro-oxidants can kill the cancer cell. And we could finally show that this is also the case when we look at the animals' prostates. This is a prostate from an untreated animal, and the red are the prostate cancer cells, which are expanded. This is only 2 weeks after or following the disease. But after 2 weeks of giving menadione, we have substantially reduced the cancer, and the tissue, the normal tissue, does not take any and show any damage. So we realized that we have something on our hands that is very, very selective, easily administered. It's a provitamin. So the big question, and doesn't have a toxicity, and so the big question for us was, does this strong therapeutic in mouse actually translate to patients? And so we realized we need help with that.
Thank you, Lloyd. And you can imagine it's thrilling to be approached by a colleague who has not only discovered a new type of cell death, but also has discovered that he can induce it in one of the most common cancers that we have, and can induce it with a drug that is a vitamin analog or vitamin precursor and is relatively self to administer. So we sat down together and we thought about how can we potentially move this into the clinic, and how can we do it so that we can do it relatively fast and in an informative way, so that we get results back from the patients in a time scale that really allows us to make decisions, and that allows us to make decisions at a cost that is really affordable as well for a small institution like us, and sponsored with a relatively small study. And the study study type that I want to introduce you to is a window of opportunity study. It's essentially where we align our research investigation to the standard of care. So you may know that many patients who have cancer, doesn't matter which one it is, prostate cancer, breast cancer, colorectal cancer, have 2 visits as they get cured from the cancer. One for the biopsy, where the true diagnosis is established, and then one for the surgery, if they are eligible for, where the tumor is excised and then actually hopefully fully removed. And so this window between having the biopsy and having the excision is often filled with therapy which is called neoadjuvant therapy. So therapy that is supposed to control the tumor, and it's time that elapses in that period that we can use for research. And so we can do the following investigation. We use this short window that you see here between the biopsy and the surgery to give the medication alongside the standard of care, and then interrogate the samples from prior to getting the medication and after having received the medication in the lab to ask the questions that Lloyd is asking. Do we see target engagement? Does the tumor respond? What is the effect on the organism? And we can do a number of clever tests on this. We take the sample out. We can look at the genetic analysis. We can look at what genes are being regulated or addressed with our therapy. We can look at the slides, as you've seen with Lloyd. Cell imaging and understanding of cell biology can be very informative. We can test, did the drug really make it into the tumor? And for that we use a technology that we have here at Cold Spring Harbor, which is called mass spectrometry, where we can exquisitely, sensitively determine if a chemical molecule has made it to the target tissue. We can do something, and I will show you some examples, data for that, which is called spatial transcriptomics. This is the latest sort of approach to look at the cellular architecture and the activity state of the cells at the same time. And we have a fantastic colleague here, John Prial at the lab, who's world class at doing that and enables our research in that way. And then we can ask, how did the patients feel throughout and what did they experience? And that's obviously done in combination with a clinical care pathway. Just want to show you some example images. You may have seen examples as images like this. This is from a PET-CT where a marker is injected into the organism, into the patient in this case, and you can trace tumors and you can trace metastases with high detail, and that can give you a guidance of where would you want to place the biopsy needle to find out where a tumor is. And here are some samples from actual sort of pre-biopsy scans. This is from an MRI scan of a prostate, where the tumor can be really marked out, and then a needle can be passed into it, and a core of this tissue can be removed before later on the organ is removed, and then we can compare that core prior to drug exposure and after drug exposure. And we can often in surgical approaches also take tissue samples from normal tissue to find out, does the drug affect normal tissue? This is actually a liver biopsy being taken by one of my colleagues, Jerry Vitiello from Northwell, during an operation where he's treating a patient with gastric cancer in this case, and we are interested in the whole body response and the host tissue, and we can take a small sample at no risk for the patient to get information of the normal tissue alongside the tumor tissue. And that's actually a sort of something that we have to rehearse carefully between the team, because time is of the essence, because the tests that Lloyd wants to do are best done at very fresh tissue, ideally directly warm tissue freshly taken. And so I'm showing you here just that teamwork is really important here. So this is the first patient where we took samples. It took us 16 minutes from taking the biopsy to freezing it down in a way that Lloyd could interrogate it in his lab. And then as the team clicks in and we sort of work hand in hand, you can see this sort of makes better work. And so we've got this down now to less than 2 minutes with the fourth patient of taking a sample and putting that sample at minus 80 degrees into the freezer so that we can do top quality assays on it. And what are the type of assays that we can do? This is not just a bit of colorful dots hitting the windscreen. This is actually a full resolution of a tiny tissue section. Here you see the whole tumor. This is from a female tumor, actually, a different disease called ovarian teratoma, where we've zoomed into this section, and this section is blown up here. And what we've done is we have here marked out in different colors the expression pattern of specific genes, and we can allocate those to those genes that might activate the immune system, the detail of which doesn't matter in this case, and those genes that mark out the immune system in something called a tertiary lymphoid structure, which is reactive immune tissue in a tumor. And so we can really see now how these reactive tissues are right adjacent to the immunogenic tissue in the tumor. And we can, for the first time, really in real time resolve the biology in the tissue. And now you can imagine how powerful this is if you could do something like this prior to giving a medication, and then after receiving a medication, where you can start building in human tissue in real time over a couple of weeks evidence of drug engagement and responsiveness within the tumor. For example, following up from Lloyd's fantastic lead of causing a new cell death in the tumor and understanding the biology of it. And so, Lloyd, I think we should talk a little bit about what's necessary for those steps. And I will make a start. As you can see from my presentation, what we need is we need a patient population, and for that we have a fantastic collaboration with Northwell Health and colleagues who are engaged in that. And then we need to obtain the tissue. And finally, we need to interrogate the tissue. And so one of the things that I thought was interesting is to cost this out a little bit maybe. So we estimate that we need about $10,000 per patient enrolled for getting the tissue and about $30,000 to interrogate it deeply. And because we can compare and contrast prior to taking the sample and after taking the sample, we often get informative results from as few as half a dozen to a dozen patients. So if we were aiming for 10 patients, we would probably get a long way with $250,000 to $300,000 just to run the study. And I want to highlight at this point that for example, the funds that you make available to Bruce are so essential, because to get funding for a study like that from public research funds or from the government is impossible because there's no funding mechanism for it. But if we have funding available from the unrestricted funds at the lab or through generous donations, we can actually really get this going. And then we need the drug, and you have some thoughts on that.
We need the drug, yes. So we were able to find somebody who can actually make that drug. So it is not something, unlike other dietary supplements, it's not something that you can just get at the vitamin shop. It is a drug that needs to be manufactured under FDA-regulated standards or good clinical practice standards, and we found somebody who does that for us, and they do it with a twist that we find would be very, very interesting, because the way we gave it to the animals was so that they were constantly exposed to a level through the drinking water. So they were constantly dosing themselves, and we just measured how much they were drinking and made sure that it actually went into tissues. So we would like to try and mimic that. And the pharmacy that could do that, they found out that they can do a slow release formulation so that the vitamin precursor, the menadione, is taken up, eaten twice daily, but then is released at a slow level so that it best mimics this constant dosing, and we don't have these excess peaks that might just be excreted instead of being used in the tissues.
Yeah, that's terrific. And so we can then, of course, as I showed you before, interrogate the samples and see, does it really make it into the tissue? And I think that's one of the things that's often overlooked in testing drugs in the clinic, which is, does the drug get to the target? Exactly. And can it engage? And Lloyd will be able to take these samples and find if the mechanism of cell death, which is unique, is induced in those samples in response to the drug. And exactly. I should have taken the remote control, because I think with this we would like to sort of conclude and thank you for not only your attention but also for your support. I want to say that I've come to appreciate the engagement with the community at Cold Spring Harbor as something very unique, having been at different institutions, and there are many people in the audience who are friends and not just supporters of the lab. And I myself have been the guest in their house, and Christine, for example, hosts a wonderful winter party that I've had many happy meals at and take my children along. And there are many other people here in the audience who are not just supporters of the lab, but are supporters of the people in the lab and welcome us to the community, and we really appreciate that. And so I want you to know that we understand that your engagement goes beyond the financial support and also goes to the community support and to making us feel welcome, and that's really much appreciated.
Yeah, I can only echo what Tobias just said. Coming back to the actual study and the topic, I have been here for 19 years now, exactly, so 2007 to today, and I can say that this is the most important and defining, I think, moment in my career at the lab. We have never been able to ask a question that we answered in an animal, in a patient, never. And we could go on finding out lots of things in the model, in tissue, in cells, etc. We could find mechanisms and everything, but it would never answer what we can be doing together. And that's a really new level, so a new era for my team. And I really, really hope that we're going to be able to do this.
Thank you.